Hello everybody, so today's video is going to be focusing on one of the macromolecules, and that is carbohydrates. So the function of carbohydrates is energy, fuel, and storage that can be used for cell communication as well as structural building blocks. So the smallest component of a carbohydrate are the monosaccharides. Monosaccharides are made up of 3 to 7 carbons. So a combination of different atoms, that is carbon, hydrogen, oxygen, in different ratios makes a monosaccharide. And the ratio can be represented by this equation, CnH2nOn, and that basically just means that N is the number of carbons, so number of carbons is equal to the number of oxygens, and the number of hydrogens is going to be 2 times the number of carbons. We can have linear forms of monosaccharides, but the linear forms of the monosaccharides contain these functional groups, either one of the following. So they can either contain an aldehyde, and an aldehyde looks like this. So it is a C double bonded to an O and then an H. And then the R is the rest of the monosaccharide. And you're going to see in the next few slides how they look like in the linear form of the monosaccharide. The other functional group that a monosaccharide can contain, right, either one of these, the second one could be a ketone, and a ketone looks like this, where we have a C double bonded to an O, and then these R groups are just those other portions of the monosaccharide, which you'll see in a minute. So the first monosaccharide we're going to look at is glucose, and glucose contains that aldehyde functional group. So here is the linear form of glucose right over here, and if you were to look at where the aldehyde is positioned, it's positioned right over here. And you can see it's the C double bonded to the O and then the H. So we can see that aldehyde right at the end of the linear form. The next monosaccharide we're going to look at is fructose, and fructose contains the other functional group that we talked about, which is the ketone. And so here is the structural formula of fructose, and if you want, you can pause the video and try to find the ketone. And if you paused it and figured out where the ketone is, it is located right over here. So this right over here is actually a carbon. That's why it's labeled 1, 2, 3, 4, 5. And so basically, since it's labeled 1, 2, 3, 4, and 5, the second carbon over here has the double bonded to the O. So these are just pure memorization of the structural formulas, but the numbers are basically indicating the carbons. So you can see that carbohydrates contain about 3 to 7 carbons, and over here each of these contains 6. So that is around the range of the carbons that a monosaccharide contains. The next monosaccharide is galactose. And galactose, just like glucose, contains an aldehyde group. So here is the structural formula of galactose in its linear form, and here is that aldehyde group. And again, this point right over here, if you look at organic chemistry, or if you take organic chemistry, you'll know that these points or these connections are basically where the carbons are located. So there's no numbering over here, but this would be 1, 2, 3, 4, 5, and the 6th carbon. So on the first carbon, we have the aldehyde group, the C double O, and then H. So when these linear forms of the monosaccharides are thrown into water, right, they form a ring in water. And this is called the cyclization process. And basically, it's when the carbonyl group will interact with the hydroxyl group. So we're going to look in a second, but basically what happens is that this end right over here interacts with this end over here. So the hydroxide on one end reacts with the carbonyl part on the other end, and it'll form this ring structure, or cyclization will occur. So the first ring structure is glucose, which again contains the aldehyde, and here is the glucose monomer, or the glucose or the monosaccharide, and this is how it looks like in its ring structure. Next is fructose, right, and fructose's ring structure looks like this, and again the carbons are labeled, so the carbons over here would be labeled as 1, 2, 3, 4, 5, 6. And over here, they're labeled as 1, 2, 3, 4, 5, and 6. And then finally, we have galactose, the last monosaccharide that we looked at, containing an aldehyde functional group, and that looks something like this. So these are the ring structures, and they're they're almost identical to the linear structure, except that now the carbonyl group on one end has reacted with the hydroxyl group on the other end, and they've kind of just joined together. So you can just imagine the looping of one end of the linear form with the other end, and that'll give you these ring structures. So now we're going to look at the isomers of glucose. So glucose has two isomers, and an isomer is basically a molecule that will have the same chemical formula, so it has the same number of carbons, hydrogens, and oxygens, but it has a different arrangement of these atoms. So to get a better understanding of this, we first have alpha glucose. That's the first monomer of glucose, and it looks like this. And you can see that, well, in this picture, it's highlighted and it's in a different color, but there's going to be something different between this glucose and the other glucose that has the same chemical formula, but the different arrangement of atoms. And in this case, this alpha glucose contains the hydroxide on the first carbon. So here's the first carbon since it's numbered, and the hydroxide is located at the bottom. So it's bonded to this carbon, but it's located at the bottom. In beta glucose, it looks almost exactly the same, but in beta glucose, that hydroxide is now located at the top of the first carbon. So the difference between alpha and beta glucose is the positioning of the hydroxide on the first carbon. Now we're going to look at disaccharides, and if we looked at monosaccharides, mono was one, and so disaccharides would probably be two different monosaccharides coming together and forming forming a bond together and forming a disaccharide. So disaccharides are two or more monosaccharides coming together, and they form something called glycosidic bonds. And so the way to do the chemical shorthand of this, or how monosaccharides are bonding together, is, let's say the first carbon on one of the monosaccharides is bonding to the fourth carbon on the other monosaccharide. When they join together, you're going to write the shorthand as 1 to 4, which means that the first carbon on the first molecule is bonding to the fourth carbon on the second monosaccharide. So this is just the chemical shorthand of writing that, and you're going to see in a second how they bond and how it looks like. So the first the disaccharide that we're going to look at is maltose. And maltose, if we go back to the slide of the different monosaccharides, and we take alpha glucose and alpha glucose, the same monomer of glucose, which was alpha glucose, and we combine two alpha glucoses together, we get maltose. And this is the general structure of maltose, and you can see that first carbon on one alpha glucose molecule is bonding to the fourth carbon on the second alpha glucose molecule. So you can see this bond right over here happening, and this bond is called a glycosidic linkage or a glycosidic bond. And something to remember actually is that if you go back to the introduction to biochemistry, we looked at dehydration, synthesis, and condensation. So dehydration or condensation and hydrolysis reactions. So in this case, water was removed, or dehydration reaction occurred in order for maltose to be formed. Because if you go back to their ring structures, there was an OH and an H. So that OH and H were removed from either one of them, and then that H2O was removed, dehydration synthesis, and that formed that glycosidic linkage and bond. So again, this is happening between the first and the fourth carbon, and this is called an alpha linkage since both of them are alpha glucose molecules. The next disaccharide is sucrose. And if you take the isomer alpha glucose and combine it with fructose, then you get sucrose. And sucrose looks something like this. And again, you can see that glycosidic linkage or bond, which was formed because of that dehydration synthesis reaction. And again, it's happening between the first and the fourth carbon. And since it's alpha glucose, this is an alpha linkage. The next disaccharide is lactose. So lactose contains galactose and beta glucose. So when beta glucose and galactose come together, the isomer in this case for glucose is beta glucose, it looks something like this. And you can see right over here, that is our glycosidic linkage or bond. Again, it's between the first and the fourth carbon on either monomer or either monosaccharide respectively, and this is a beta linkage since beta glucose is involved. So polysaccharides are going to contain hundreds and thousands of monosaccharides combined together, right? They're complex carbohydrates that are composed of hundreds of thousands of monosaccharides that are linked together. And they can serve as many functions. So energy storage is one of their main functions of polysaccharides, and that can be in the form of starch or glycogen. And we're going to look at each of these in depth. And it could also be structural support, like cellulose and chitin. Starch is a storage form of glucose, and it's used in plants for energy storage. It's made up of alpha glucose monomers that have a 1-to-4 linkage, and that means the first carbon on one glucose is linked to the fourth carbon on the second glucose. There's two forms of starch. The first is amylose, and amylose is basically just a straight unbranched chain polymer of glucose units that's made up of alpha 1-to-4 glycosidic linkages, and it's basically 500 to 2,000 molecules of these glucose. Amylopectin is many or linked or branched amylose chains. So if you were to combine a bunch of amylose chains, you would get amylopectin. And these are made up of alpha 1-to-6 linkages at these branch points right over here. And millions of molecules of glucose combined would actually make amylopectin. Glycogen is also an energy storage polysaccharide, but it's in animals. It's very similar in structure to amylopectin, except it's even more multi-branched. And it's made up of alpha 1-to-4 linkages again in the straight chains, but then alpha 1-to-6 linkages in the branches. So here we can see that alpha 1-to-6 linkages at the branch points and alpha 1-4 linkages between the straight chains. And glycogen is going to contain helixes. It forms helixes that are held together with hydrogen bonds, and that forms this complex structure. The practical implication of glycogen can be seen in insulin and glucagon. So looking at glucagon, it maintains your blood sugar level when blood glucose is way too low. So when it's low, then glycogen is converted to glucose by slowly taking off glucose monomers from the glycogen and distributing them into the blood, and that increases the blood glucose level. But insulin, on the other hand, maintains blood sugar levels when blood sugar is too high. And so in this case, glucose is added onto glycogen to store it as glycogen. So you can pause the video and look through this flow chart to kind of get a better understanding, or just look at it in a more pictorial view. Cellulose is a major component of cell walls, so of course it's used for cell support. It's made up of beta glucose monomers with 1-to-4 linkages. It's long and straight, and it has unbranched chains with large numbers of polar OH groups. And in this case, every other glucose is upside down, and this allows for hundreds of thousands of hydrogen bonds to form between the chains. And that allows for that rigidity in the cellulose molecule, allowing for it to be for support. So here we can see every other glucose molecule would be upside down if we were to straighten this line out. And that allows for hydrogen bonding, of course, and again, relating to the rigidity. And it's a dietary fiber, meaning it can't be digested since we don't have enzymes to break it down. But remember, it's very important to have in our diet. Chitin, the last polysaccharide we're going to look at, provides support and protection, and it's long, straight, unbranched chains as the polymer. It contains beta glucose with 1-to-4 linkages, and it's also made up of N-acetylglucosamine, which is NAG. And so this is the subunits of the sugar contain this functional group that has nitrogen in it. And every other glucose is going to be upside down. So here we can see every other glucose is upside down due to this line. If we were to straighten it out, we would see that this would technically be upside down. And here's that NAG functional group containing that nitrogen on the glucose molecules. And obviously, chitin is used for support and protection. And an example would be in like the exoskeleton of beetles and other insects.